actually want to start this. Okay, ready.
Hi everyone, welcome. Thank you for joining this Bellevue College Earth Week event as we celebrate the 50th anniversary of Earth Day. We still have more events in store, so please make sure you check out the full schedule at our website, bellevuecollege.edu/sustainability. You can also find recordings of our previous events there too. So before we start the presentation, there's a few things to go over. First is etiquette. The event is being recorded, so please be respectful and polite. All participants' microphones are muted and cameras disabled. If you have any technical issues, need our attention, or like to ask any questions to our speaker, please use the Q&A box to communicate. That box is located in the top right corner of your screen, and there's a little Q&A icon. Messages sent to the Q&A box will be first reviewed and then posted, so you might not see your question in real time. The video is also on a few second delay too. All questions in the Q&A box will be asked in the middle of the presentation and at the end. So make sure you stick around till the end if you want your question answered. If you need any live captioning and don't see it already on your screen, you can go to the icons button on the top right corner and click that and then click live captioning. Lastly, after the presentation, please fill out our survey and let us know how we did. It's quick and it helps us make events like this possible in the future. We'd really appreciate it. Look for the link in the Q&A box. We'll be sending it out there. All right, let's get started. So my name is Alyssa Gordon. I'm the program manager for the Office of Sustainability, and our team coordinated this event for you all. Today we are thrilled to be joined by Dr. Tierra Moore. Dr. Tierra Moore is a postdoctoral scholar at the Nature Conservancy of Seattle. She has been featured in the Seattle Times, Chicago Tribune, Index Journal, Baltimore Sun, Phys.org, Aslo Bulletin, ProQuest, Daily Bruin, and Reddit. Tierra received her BS in biology at Winthrop University in South Carolina. She completed her MS in biology with a concentration in environmental science at Hampton University in Virginia. She earned her PhD in ecology and evolutionary biology from UCLA. Dr. Moore has conducted research on the Chesapeake Bay, Carpentera Salt Marsh, Upper Newport Bay, and the Ellsworth Forest. Internationally, she has conducted research in Bali, Indonesia, and French Polynesia, where she has explored various topics such as the effects of microalgal decomposition on sediment biochemistry and the microbial community, using environmental DNA techniques recently developed to assess the biodiversity of entire ecosystems with only one soil sample. A long way from a little town on the East Coast, Dr. Moore hopes her research inspires others to cherish the small things, especially dirt. I'd now like to welcome Dr. Moore to our event today. Welcome. Hi, Dr. Moore.
Hi. Hi, everyone. How are you? I hope y'all are having a great, I guess, just Earth Week, Earth Month, Earth Day. So I should now share my screen, right?
Yes, please.
All right, perfect.
There we go. Perfect.
We're good?
Yes.
All righty. So yeah, thank y'all so much for just joining today. So like you said, I am Dr. Tierra Moore. I do use she, her pronouns. And I just want to take 1 second to acknowledge that we are on the Duwamish land. So just take a second to just acknowledge the land as we are thinking of Earth Day and Earth Month. So we are grateful that they took care of this land for us. Thank you. So for my talk today, the title is Dirt DNA, Establishing a Thermometer for Fighting Climate Change. And let me see, make sure I can advance these slides. Okay, and so for my talk, basically, I'm going to be focusing on how we use this environmental DNA or EDNA data as a tool to be able to really assess what's going on in our changing environment. And then after we get this data and we make these assessments, we then use these these data to inform our management practices to be able to kind of prevent future impacts. And so that's kind of the flow of the talk today, and hopefully the goal of you say you went to this talk and that's what you got from this. But before I get started, I do want to just talk about myself for a little bit because, you know, first I want to talk about me. Just to give you a little background. I know this is, you know, a college, and I know that some of you all may be, you know, trying to figure out where you are in your own journey and what you're doing with yourselves. So I just want to talk a little bit about my background and just to give you an understanding of who I am as a scientist and how I look at the world and in my research. So from the introduction, maybe you can tell from my accent at this point, I'm originally from Greenwood, South Carolina. So this really, really small little town in the country. And so, you know, when I went to undergrad, I went to school right in, you know, in South Carolina, and I majored in biology and actually was pre-med, and I thought I wanted to be a pediatrician. And I realized over time that maybe I didn't like kids as much as I thought I did. And so I was like, oh no, this isn't going to work for me. So what am I going to do? And so there was, you know, a series of classes. That's the beauty of being in college, especially at a liberal arts school where I was at. You know, you can just take a bunch of random classes. And so where there was a course called Tropical Ecology. And to be honest with you, I signed up for it because they were going to Costa Rica for spring break. So I was like, oh, free trip. And so there on this trip, we're on a boat and we're collecting water samples and we're doing these experiments. And I was like, wow, this is a real job. Like, y'all getting paid to be here right now. Like, wow, how can I get involved with this? And so I found out about the whole world of marine science. And so from that trip, from that one experience, I changed, I guess, the trajectory of my education. And, you know, I started focusing on that and I applied to grad school. I found a master's program that was, had a 2-year program for marine science, for environmental science. And so I applied to that program. And so this is, if you think you are not qualified for something, just apply and ask. If you're changing paths, definitely just still do it, because I wrote in my personal statement, like, hi, I took one class, please accept me. I really, this is my passion. And so they did. So I went on to do my master's in environmental science at Hampton University in Virginia, which is a historically Black college and university. And so I did research on the Chesapeake Bay and the nutrient pollution that comes from the watershed there because it's so human dominated. And so from that experience during my master's, I felt confident enough that I could keep going. And so I went on to do my PhD at UCLA. And so from that work, at that time, I continued to do research on these human impacts in the environment, and particularly in the marine ecosystem. And at that point is where I got my training and I learned about this world of environmental DNA. And so now I'm doing a postdoc in partnership with the University of Washington and the Nature Conservancy. I'm really diving more into the field of environmental DNA, and I'll talk more about that project throughout this presentation. So that's just a little background. This is me saying I am a marine scientist by training, but I'm going to be talking about genetics and molecular ecology. But, you know, just letting you know that's my background. Alrighty, so let's get started. So a lot of my research that I talked about previously, and what basically will keep me employed probably forever, is us humans. We are causing a lot of major environmental impacts globally, daily. And so things like destruction of habitat, we have, you know, deforestation, we're wiping entire ecosystems because we need, you know, more, I don't know, Targets and Walmarts and things like that. We need, there's logging practices. So we destroy these ecosystems and then we displace wildlife. Over harvesting is also another major human impact on the environment, because we all do love our salmon. I've learned since I moved to Seattle. But of course, there's other fish, our sushi, every time we want to eat these organisms, we're fishing at rates that are so large that we're seeing these ecosystems die down. We're removing them from their communities, and so there's other organisms, fish, that depend on them, and so they're not able to sustain these ecosystems because we're fishing at such large amounts. Other things, the big one that we've all heard of, climate change. And this is something that's really interesting to think of over this last 50 years of change since we're thinking of Earth Day. This is, a big increase in climate change is due to the large amounts of carbon dioxide that is in the atmosphere, a major greenhouse gas. And this is due to these emissions from transportation, from big, large businesses, manufacturing practices, things like that. And so all of these this is because humans are causing these practices, and then it's leading to this excess carbon dioxide in the atmosphere. So a major byproduct of this climate change that we're seeing in our oceans is ocean acidification. And this is just really interesting to think about this example. So if you look at the top, I don't know if you all can see my mouse, maybe not, but at the very top, you'll see that this is an intact shell. And so ocean acidification basically occurs when we have this excess carbon dioxide that's in the atmosphere, and everything in the atmosphere is absorbed by the surface ocean. It's interaction, right? So there's so much carbon dioxide, it causes the ocean for the pH to decrease. It gets acidic. We have a lower pH. So imagine you have this shell up here at the top that was just minding its business, in an intact shell. And then for this experiment that we have an example of here, it shows the shell exposed to pH conditions expected to be in 2050. So that's not even 50 years from now. So you can see that the shell has completely started to deteriorate. So just to bring this home, you can imagine, as we are right now, sheltering in place in our homes, if you were just sitting in your home like the shell was and someone just started to tear down the walls, that's exactly what's happening to this organism. It's there and its home is just slowly deteriorating around it, dissolving because of the acidification. Another byproduct, another major human impact is nutrient enrichment. A lot of the fertilizers and things that are used for our agricultural practices are highly enriched with nutrients. Every time there's a rain event, all those nutrients are washed into our watersheds. And these have a lot of impacts. They can cause major fish kills. They cause these things known as these macroalgal blooms. And so these blooms form because of these largest amount of nutrients. And so these nutrients get into the watershed because of this agricultural practices, because of these excess fertilizers, because of waste dumping, because all these things that will cause these nutrients to get into the watershed. And then we'll see this byproduct of being these macroalgal blooms. And so when I found out about these different, I guess, human impacts and how they were adversely impacting the environment, I was really just like, wow, like this is so much that's going on. What can I do to help? What can I do? How can my research be impactful? And so when I started my PhD at UCLA, I actually had the opportunity of working with my own macroalgal blooms. So here's me in the field, and this is in Southern California. And so our bloom in Southern California, our estuaries are very shallow. And so you can see that green, that is macroalgae, similar to the picture I showed you before. And so we have this large bloom that comes in because of this nutrient pollution, and it basically takes over this watershed. And so I was really interested in the dirt. If you haven't noticed yet, my theme is dirt. And so I was already interested in the sediment and what it was going on, because there's a whole vast community that lives in the sediment, and that there's just so much that goes on in the sediment. Yeah, that's a whole separate talk. Anyway, so I was already interested in the sediment, in the biogeochemistry. So what's going on with the water and the sediment interactions? And now we have this biomass, this macroalgal bloom that's coming in and interfering in this whole environment. So I was, completely interested in that, and what was happening in that interaction. And so by me becoming interested in this macroalgal bloom and what was happening with the sediment biogeochemistry, and I guess I was answering the chemical question, but then the biological question intrigued me. So what is the bacterial community? What is the microbial community in this ecosystem doing? And so by asking that question, I opened myself up to a whole larger field, a larger experience, and the whole, I guess, purpose for this talk today is environmental DNA. And so what is environmental DNA? And basically it is just the DNA that is collected from an environmental sample. And that sample can be water, air, it could be, of course, soil. And so as these organisms are just interacting, living in their ecosystems, they are shedding skin, fur, plants are leaving leaves, pollen, different things. And that all has genetic material. And so basically we can collect these samples, extract the DNA with a series of lab work, a series of steps, and then that can give us a real-time kind of snapshot of what's there, who's there in that community. And so that was really cool for me because I knew what was happening in the ecosystem with the macroalgae and all that, but I didn't know, that invisible community. I couldn't really see that. So it added a different layer to my research. And so from then, and this is, I guess, another talking about slipping in the journey story, is when you are a scientist, if you think about doing research as a student, we have the opportunity to go to conferences and things like that. And I would definitely suggest that you do that if you have the opportunity, if the university has any type of funds, if you're interested in a professional organization that has student travel grants, things like that. I would definitely say to apply and go to those events and things like that, because it is really important for you to network and really get your research out there. And so that's what leads me again to this next part of my talk, is that, I was talking about my research at different conferences and things like that. And it's where I met my current supervisor. And we're talking about this new tool, this environmental DNA, of how we can take a small sample and really get so much information and data for our research, and how this could be really, really important. And so, me being new to the molecular world, me about to graduate with my PhD, I'm excited. I'm excited that he's excited, because, when you are a scientist, you don't know if anybody's going to care about your research at all. So it was cool to say. And then we had conversations about it. Well, how could this research be applied to, a terrestrial setting, to a forest? How could we take these soil samples in a forest and assess the, biodiversity? Can we really understand what's going on in the ecosystem there? And so, being a scientist, I'm sure, yeah, that seems reasonable. Like I could definitely see the methodology similar to what I have done in the marine system with sediment. Sounds good. And so the question, as we continue to talk about this, well, could you do it? Could you, do this type of project in this forest with this EDNA? And so I'm thinking in my head, oh, well, I'm a marine scientist, that we're talking about how I did this in the marine world. And so again, thinking about this little tidbits of advice, I guess, don't be so tied to what you really, what your plan is. As you can see, I've already talked about the twists and turns in my journey a little bit, but even in the research that I've spent my time working on, I was able to adapt it because, as we were talking about this conversation, if I could do this same research that I'd done in the marine environment and terrestrial, I was asked one of the most profound questions and that made me say, okay. And he asked me, well, dirt is dirt, right? And so I think that's really interesting. And I think that's why EDNA and this process that I'm going to talk about for the next half of this talk is so important, because in the simplest concept, dirt is dirt. So we have a very powerful tool where we can take any type of dirt, soil from any type of ecosystem. So it takes out that, I guess, that ecosystem aspect, and we could apply these same methods and these same mindset about the the results and the data for the application globally. So really, we can really, if this method, if we say it, it's as important as we're saying, and that we get the data that we really need, we know that, okay, it could be used in the marine system, it can be used in the terrestrial system, it can be used in any system. And that's really important as we're thinking about, the 50 years of change that we've seen over time. And then we think about protecting and conserving for the next 50 years. And so I think that's why, marine scientists in my heart, thinking about how we can use this particular method and the novelty of it really intrigued me. And of course, the profoundness of this question, dirt is dirt. So I think, let me see, I'm going to start talking about my next project. I don't know, we had talked about answering some questions. I don't know if there has been any questions so far. I can keep going.
Yeah, we haven't had any questions yet, but everyone just remember that you can click on the Q&A box and put your question in, and we'll ask Dr. Moore. Thanks.
Great. All righty. So all right. So we know dirt is dirt. We know that I'm doing environmental DNA research. And so for my project now is in Ellsworth Creek. And so this project is down in Nassau, Washington, down Long Beach area. So down, again, I'm still learning the area myself. And so this forest basically is an 8,000 acre old growth forest that was clear cut years ago. So clear cut, basically you remember that picture that I showed you at the beginning. That's an example of forest being clear cut. And so in order to protect and restore and conserve that land, the Nature Conservancy purchased it in 1999 and started restoration in 2004. And I put a little clip to the Seattle Times article that was featured for the project that I am talking about. If you want to get a little bit more in-depth information, it was a good story. But yeah, so this is just the background of the project. And so if you think about it, so we have this forest that was clear cut, and then we have an organization that's goal is to conserve nature, by this place, and with the goal to restore it. And so I guess the question that you immediately ask is why? And I think in order to understand the reason why you need to restore a forest is truly understanding the importance of trees. And we think about climate change. I know there's been a lot of talk about tree plantings and things like that, and all these things we can do. And of course, there is a lot of other things that we can do to help mitigate climate change, but trees are highly important. And so trees provide habitat, of course. They provide nutrients and food for a lot of organisms, including us. They provide resources. And most importantly, if we're thinking about climate change and our environmental impacts, they provide carbon sequestration. And so I'm going to see if we can get an answer here. But so I have two numbers on my screen, 88 and 44,000. So I want to see if anyone can guess what they think these numbers represent. What do you think 88 represents, and what do you think 44,000 represents? If no one knows, we can just I'll throw up the answers.
Feel free to answer the question by typing it in the Q&A box.
I feel like I should sing like the Jeopardy. Do, 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 do. All right, guys, you can't say I didn't try. I was planning to be in person, wanted to interact a little bit if I could. It's okay though.
We did get one answer.
Oh snap, what do we have?
88 species.
88 species. So, okay, it has to do with carbon sequestration. So, not 88 species, but I will go ahead and tell you all. So, when we're thinking about again, climate change, I told y'all, was because there's a lot of carbon dioxide in the atmosphere. And so the reason why trees are so important is because they can sequester this carbon. Basically, they can remove this carbon from the atmosphere. And so this 88 is important because it is the 88 is the amount of pounds that the average tree removes from the atmosphere per year. So that's cool. We know that, okay, if we plant trees, they can remove 88 pounds of carbon. But if you look at the other number, and that's what's really important, and that's why people are like, oh, trees aren't going to save us. But they could help. But the other number, and this is really important, and that's why it's really important to think about our carbon footprint, is on the opposite side of that is 44,000 pounds is the average amount of carbon that humans emit per year. So if you think about it, we have a tree that can store 88 pounds, so yay, tree. But then we have us who are just shooting out emitting pounds of carbon. So it is off balance. But again, we're talking about one tree. So if we have forests like Ellsworth, old growth forests, who are capable of sequestering tons of carbon.
155 moment mark. So we can just, touch base.
I don't know what's happening.
Just to see how everyone's writing session or work session went.
I am so sorry. Okay, okay. Oh, that was so, that made my heart flutter a bit. Okay, sorry. I don't know. That was weird. I guess a video just popped up or so. I don't know. Anyways, so yeah, so humans are emitting though 44,000 pounds of carbon. So we can say if we have like here's where my thought was going. If we have an old growth forest that's able to store tons and tons of carbon, then that's good to help us mitigate the impacts from the human emissions. Okay, so with trees though, we have to, so in order for trees to be healthy, we have to also understand the importance of soil, my favorite thing. So like I said, similar for trees, we also have soil, and they provide the foundation for trees to be able to grow. And so soil is important because of course it also provides habitat, it provides nutrients as well, and then it also has long time carbon storage. So I do have another question. I don't know if we can try this again. So now we know the theme of the other. So the first number is 3,000 billion, and then the second is 2. So thinking about carbon storage and soil, what do y'all think these numbers represent?
Remember, we are on a few second delay as well, so let's give them a few more seconds this time.
Thanks for reminding me. $3,000 billion and 2. We think about soil carbon storage. Thoughts? Thoughts?
We have in fungi or a question asking 1,000 billion?
1,000 billion. Okay, So for this one, this is also about carbon storage amount. So thinking about the trees, we said they were able to store 88 pounds. Meanwhile, we're emitting 44,000 pounds. But the importance of soil, soil is able to store 3,000 billion metric tons. So that's the whole global carbon soil storage. And so there's 2 times more carbon in soil than in vegetation and air. So if you think about that, you see soil is really important if we're thinking about getting carbon, removing carbon from the atmosphere. And that's what's really important. So we have the reason for it to be a great foundation. It needs to be healthy to be a great foundation for our trees that are able to sequester and store carbon. But the soil itself is also really important in storing carbon. So trees get a lot of press, but I'm telling y'all, soil is important too. Dirt is important too. So just with that kind of background and the reason why we're worried about conserving and restoring this huge forest is really because this trees and soil are so important. So with this, I guess, background of EDNA and everything that I've talked about, for the project that I'm working on now, we kind of are asking 2 major questions. And with EDNA, with the data that you receive from it, basically, these are the types of things that you can ask. And so our first major question is, what is the biodiversity of the ecosystem? And you can think about this being important is because, okay, if you again remember the picture from the beginning, you think, oh, we had an ecosystem that was completely wiped out. So the wildlife was misplaced. They had to go elsewhere. All different, the commute, all these different ecosystems were disturbed. So when you're restoring an ecosystem, of course, it would be great to know if the wildlife community is coming back. We want to know, like, oh, is this helping? Are we bringing this ecosystem back to where it was? So that's the first major question that we ask, and we can do that with this EDNA analysis. And then the second major question that we're asking is, what is the makeup of the soil microbial community? And I kind of talked about this a little bit when I was talking about my past research at UCLA, is that, the soil community is really important. There's a whole ecosystem that's going on there that's really running the show that doesn't get a lot of press. And so a lot of that is that the soil microbial community will promote, a healthy root system for trees to be able to establish and grow roots and get the nutrients that they need to be able to grow. So understanding what the soil microbial community looks like will be able to help us to see how the tree's growth projection is. So that's just 2 of the questions that we're asking. So here are our sites. This is a map of where we are doing this project. So like I told you all, there is it is down in Ellsworth, down in Nassau. And so with these, what the map shows is basically the type of sampling that we took. And so our question just was, we knew that we have this forest that we wanted to restore. Basic forestry management treatments will just say, okay, let's, we have like active treatments where it says that we are logging, removing some trees from the area, but they don't have any roads. Passive means there isn't any logging and there isn't any roads. And then of course there's a control, so we can see what's kind of going on in the background. And then we also have samples from old growth where the area wasn't clear cut, so we can just have a baseline determination of the ecosystem, and then the area that was clear cut, so we can see what happened when the destruction occurred. And so this is really interesting thinking about with the EDNAs, because okay, if we see a difference in these different treatment areas, then we can go ahead and say, oh, now this will help to inform our management. So maybe we see a more robust reestablishment of wildlife in our active treatments versus our passive. So then we would say perhaps, that we should change our management to active throughout. So that's the type of just real-time use of this data, if that makes sense, hopefully. So, picture time in the field. So here's just couple of pictures of me in the field down in Ellsworth. So you see, here's an example up at the top is some of those old growth trees that are still in the forest. So that's the type of capacity we would love to get back to. So that tree there would definitely be storing more than the 88 pounds of average, you know. Whereas in the next picture, where you see the smaller trees, those are those, you know, those are the 88 pound trees. Those are that smaller capacity trees. And that's where we have the restoration going on. So these are trees that were more recently planted after the destruction. And then this is just me using my very high-tech and fancy Slurpee straw that I use to collect our eDNA samples. And basically we do this because we do need to keep a level, level of sterileness between the samples. And so basically I can go out, literally just open up the Slurpee straw, open up a new Slurpee straw, scoop up the sample, put it in the tube that you see down at the bottom. And then just close it back up and then put it in that little bag. And then I just have, a bunch of Slurpee straws in the field with me. And so I don't have to worry about like sterilizing, getting out alcohol and all that in the field. I can just ploop, ploop, ploop and keep it pushing, because it is a bit much when you are doing field work and trying to collect a bunch of samples. As you can see, we're in the forest. So let's see. So in the field, we collect these samples. Oh, so just about the sampling. For the two different questions, we are doing two different, sampling schemes. So for the first question, just what is the biodiversity of the ecosystem? We basically want to know what's just around, what's happening on the surface soil. So for that, we're collecting surface soil samples. So that's me there just collecting the scoop of dirt on the top. And then you'll see in the middle picture where we're answering the question, we're asking the question about the soil microbial community. We are using this soil probe, and we actually get a little bit up underneath the surface to get samples from there. So there's two different soil depth levels. So then after, exciting time in the field, we then take these samples to the lab. So here's me taking my samples out when I get into the lab, and then showing you my Slurpee straw, my sample. Then through a series of steps, we basically chemically wash the DNA and extract the soil sample and extract the DNA, and we end up with this clear liquid that you see in the middle picture. And then we tag it with these universal primers through our PCR process, or polymerase chain reaction. And so this step is really interesting, and what makes this whole process just really cool is that you have this DNA, as you see me looking at it, in this clear liquid, and it's all the DNA that was there. That was just little every fragment that we were able to extract from the soil sample. Then through the PCR process, you tag it with these universal primers, that encode for specific regions, that then encode for animals, bacteria, fungus, birds. There's so many primers that you can then use. And then with that data, once we tag it with the PCR, with the primer, and then we see if the tags actually attach to the DNA through this cool process called gel electrophoresis. And we're going to watch a little short video about that next. And then after that, we see that, okay, it's tagged, then we send it to be sequenced, and we run it against this whole taxonomic database, and that will tell us what was in that sample. So that was definitely a shorthand version of everything that happens in the lab. And then, so next, I do have a video. I think the the goal is to get you all to watch it on your own. I will be playing it here, but you can't hear it. But the video is basically, it's the gel electrophoresis process. So it's after we do the PCR and we put all these primers on all these tags, and then we basically see if the tags are attached. And I'm very proud of this video. I made it myself. I added sound effects, so that's why you have to listen to it with sound. But yeah, so I hope you all take this second to just watch the video, and yeah, we'll be back. And I guess once you post the link, I'll just like play it, but definitely listening to it on your own.
The link is now posted in the Q&A box.
I'm Dr. Tierra Moore, and welcome to the lab. Wait, is it? Can you hear it? Oh, wow. How exciting is this? So today in the lab, we're going to be doing a process called gel electrophoresis. Show you a sneak peek. We loaded our gel, we started to run, and we have to keep it covered because the stain that we use is light sensitive. But come in, look what we got. So you'll see that the gel, you'll see the DNA fragments, they are actually starting to run. So hopefully when we put this under UV light, we'll actually see those primers that we put onto our DNA fragments. Yay! All right, our gel is ready to run. So I just turn off the gel rig, unplug it from the machine. Oh my goodness, come look at this gel. So here are all the samples that we just ran, and we see that the DNA fragment moved, something happened. So hopefully we'll see our amplification. So I just take this whole rig, take it over to our imaging machine, which uses this UV light to get us a photo of our DNA. So I'm going to move it around a little because we don't want any air bubbles. Close the imager. There's still air bubble. Make sure that it's on the screen, on my computer screen. Close the imager so you have one good one off because we don't want to have any of that nasty styler on our imaging machine. And then run protocol. All right, guys, this is the moment of truth. I could have just been here for about 3 hours doing that. Wouldn't that be so sad? Here we go. Bam! Hey! So this says PCR worked, everyone. Oh my gosh. So as y'all can see, I cracked myself up in the lab because I spend a lot of time in the lab, but that was the end of a very long day. So we spend the time tagging these primers to it. You never really know what's gonna happen. Like, oh, and so this was particularly good run. And so you see that, okay, we tag these primers, and then we can then send this out to be sequenced and analyzed to get the species that were present. And we end up with this biodiversity census. So this next slide that I'm showing you is from my research that I talked about at UCLA. Unfortunately, I don't have any data from the Ellsworth project to present, but I did want to give you all an example of the data that you do get from the eDNA analysis. And this is actually really cool. So if you remember what I was talking about in the estuary there, we have these macroalgal blooms that come in. And so we see that it changes, water clarity and then the bacterial community. And so basically I had 3 different sites in that estuary, the head, the middle, and the mouth. And this is the results of my eDNA data. And so it was really cool to see at the middle site, you can just look really quickly without just looking at the colors without knowing anything about bacteria, you can see that the middle site had a largely different bacterial community than the head and the mouth. And that was really, really cool because I knew from my chemical analysis that it had a completely different water quality as well. And so I was able to relate this water quality analysis with this bacterial community. And so that was because of this EDNA analysis. So this is just an example of the type of data that we will get in the Ellsworth project. We'll have this biodiversity census. We'll be able to say, oh, in our active versus our controlled treatments or in our passive treatments, this is the type of biodiversity we're seeing in these different ecosystems. Something else that we'll also see is that we have common organisms that are usually identified. And so this is really cool. We did do a, this is just from another project that I did as well. And we found, we were doing, I guess, some matching projects. And so we were doing the EDNA analysis with actually what we were seeing out in the field visually. And so we got confirmation that we actually saw this in the field and pick this up with our EDNA analysis. So this is just cool that we can see these things that we do see in the field and that human surveys do catch, but then we can also see the bacterial and microbial communities that humans cannot. So what? So after all of that, what is the purpose? What is the point? What does this really mean for 50 years of change, for climate change, for Earth Day, for our environment? And the important things that you get from this EDNA assessment in this tool is that we can see in real time this wildlife community reestablishment. So if you think about the the Ellsworth ecosystem and things like that, it would take up forever for humans to be out there looking around, doing these surveys, maybe setting up these cameras and things like that. But we can just go out, collect this soil sample, and really see, oh, if what our practices are is working actually in real time. So that's really cool. We can have a real-time biodiversity assessment. And so that's super important as we're thinking about our changing communities and how things are happening rapidly. The second thing, the so what for our second question, is the soil health assessment. And this really is important for us to be able to make these informed restoration efforts. And I'll bring up this picture again. And so it's really important to understand the health of the soil. And I didn't talk about this before when I had this picture up, but this is the best picture to hopefully really drive this point home. If you look at this picture up here at the top, you'll see healthy soil and unhealthy soil. So if you're looking above ground, you see two trees. And you don't really know, you just see, two trees, like one of, they're both growing, to the untrained eye, both just looks like a tree to me. But now that you see them side by side, you see that the one with the healthy soil is more lush, it's larger. So this tree in particular will be able to store more carbon and be more impactful. Whereas this unhealthy soil, there's a tree there, but it doesn't have the same carbon storage capacity, doesn't have as much trees. And so this is what we're trying to check for in real time, so we can say, hey, we know this soil, what this soil is doing. We could change our management practices right now in order to help mitigate these practices. So again, I just want to drive this home for the carbon storage, put that back up there. That is 2 times more carbon in soil than in vegetation and air. So as I'm getting to the end, as we're thinking about, again, this changing environment, Earth Day, and things we can do to make a difference, I hope that if I haven't convinced you of anything else, I hope that I've convinced you of this. Dirt is life, right? And so that's just funny because it's something that we walk on, but it's really a climate change solution that's right under our feet. And so with that, I would like to thank you all for listening to me today, for taking time out of your day to join our talk, and I will take any questions. And feel free to contact me at the ways below on my screen.
Thank you so much, Dr. Moore. We really appreciate you taking the time today to, talk with us and present your research. I found it so inspiring personally to hear about your journey and your amazing research that you've been working on. So yeah, now we will ask questions that people have put in the question and answer box. All right, so first question is, do older trees sequester more carbon, and do certain species of trees sequester more based off soil type?
So that is a really great question, and I think that's the exact question that we're trying to answer in Ellsworth. And so I want to say I have my hypotheses as a scientist, but I'm not 100% sure. And so that's really the rationale behind the project, because we have soil samples, and I didn't go into this, so thanks for calling it out, but we have soil samples that we've collected from different tree types, so maybe cedar, hemlock, to kind of answer at this question. And then we also have soil samples from different tree ages. So we're working in partnership. I'm working in partnership with the actual forester, someone who knows about trees, not someone who's just out collecting dirt. And he's actually, he went out with me and aged these trees. So we have different ranges of tree ages. So we're hoping to answer that question with this data and with this project.
Great. And so this kind of ties in with that question as well. Do specific soil samples store more carbon, i.e. loamy, dry, etc.?
Right. So there is different, the short answer is definitely yes. So there's different reasons for that. A lot of it that I talked about before was soil health, but then there's just also different soil types. There are like coarse soil, fine soil that are able to just store more carbon for sure, yes.
Great. Lori asked, where are the estuaries in Southern California? I'm originally from San Diego County, where we have coastal lagoons.
That is so funny. So I got caught out a lot actually for using estuaries coming from Chesapeake Bay as a large estuary versus a coastal lagoon. I say that lagoons are types of estuaries, so I can get away with this. But the estuaries that I was working in particularly was the Carpinteria Salt Marsh and the Upper Newport Bay. So you're probably familiar with those, but maybe you wouldn't consider them estuaries, but I do.
All right, Lydia wanted to know if you were using actual Slurpee straws in your research.
Yes, actual Slurpee straws. And people always ask me, did you go and like take them from the 7-Eleven? No, we did buy them. So as we know, the great and powerful Amazon has everything you need. So I just went and typed in Slurpee straws on Amazon, and they're individually packaged, they're sterile. So every time I go on the field, I just pop one open and scoop it up. So yeah, they are available for purchase in bulk at Amazon.
Like everything. Okay. Madeline asked, how many people are on the Ellsworth project? What are their backgrounds and how do they help your research? What's the process of one of your samples? What's the process if one of your samples does not match during the identification process? Is it possible to once again obtain that specific sample?
Yes, so our team, so it's I guess for as far as the EDNA, like I'm kind of like running that project, that aspect of it, but then we do have like a whole team of other scientists. So we are in partnership with the University of Washington as well, and that's where I conduct the lab work. And so like I mentioned, we have a forester who's in charge of like the tree aspect and the carbon aspect of the project. We also have a actual soil scientists, so we took soil samples as well to do like soil analysis, like the chemical analysis. So for the person that was asking about soil type, we have collected those samples as well, and that's her project and what she's working on. We also have people, what else? To help me with thinking about the genetics and the eDNA aspect. Yeah. So I think that's that first part of the question. And then the second part was if a sample doesn't match. Mm-hmm. I'm not really sure what match means because basically what we're doing is I don't know what I'm looking for, to be honest. So that's why we're using universal primers. So it's almost like an expedition. So we're saying, hey, we have the sample, there's DNA there, and we tag it with a series of universal primers. And these universal primers basically go on like a search mission and they bind to the DNA. And so and then I run that data against a database, and that shows me things that are there that are known, that have been identified. And so maybe this is the question. So if we have DNA that's in the sample that has not been identified before, which is possible, then that will come back as like not identified. It'll just say like NA or something like that. And then so that's more work on my part. Like, so now that genetic sequence is now in the database though. So if anyone else does sequence it, then they'll work on like the taxonomy of it. So that's kind of how like the database aspect works. And that's how we're even able to do this type of project, because of the geneticists from past that have been constructing these databases, that I can now take my samples that I literally don't know what's in them, and I can run it against their database and see what's there.
That's amazing. Great. And so the last question I have for you, somebody asked, how can I help?
Oh, that's such a great question. That's always a great question. And I'm always like, I should have like so many things. How can you help? How can you help? How can you help the world? How can you help me? How can you help? I think for a lot of the things that I was talking about, I mean, just being more conscious of our carbon footprint, you know, just how we're getting to work, how we're driving around, how we're getting our food, are we, you know, buying sustainably, are we doing these things? And then also, who are we supporting in policy? Who are we voting for? What are those people supporting? Are they supporting things like fracking? Are they supporting things like cutting down these large, you know, natural ecosystems? Those are the type of people that we don't need controlling our environmental funds and our budgets. So I think that's really important, being mindful and making conscious decisions. And I'm not saying everybody needs to go vegan and all that, because I consume meat. But it's about, again, like how was it bought? How was it caught? Was this one of these like batch catches, like the picture I showed you, or was it, you know, caught sustainably? How are we doing? Like if I'm buying flights, I don't know, is it a completely full flight? Things like that that we can actually take charge of. But on the larger scale, because this climate change is due to such large like businesses just doing some messed up things. And so basically finding ways to not support those is really what we can do as a people, because we have voices and we can fight against the big people.
Thank you. Great answer. All right, well, that will wrap it up. Thank you, Dr. Moore, so much for joining us, and thank you to everyone else for attending this presentation. Make sure you check out the rest of our Earth Week schedule on our website, posted in the Q&A box, and have a great day, everyone. Thanks.
See y'all. Thanks.